Hello everyone. We have previously talked about the mitochondria, where we discussed glycolysis that takes place inside the cytoplasm, where glucose is converted into pyruvate, and then pyruvate is shuttled into the mitochondrion, where it is oxidized to acetyl coenzyme A, which enters into the citric acid cycle, and from the citric acid cycle, two electron carriers, NADH and FADH2. They carry electrons to the oxidative phosphorylation, to the electron transport chain. So electron transport chain and chemiosmosis together, this is what we call oxidative phosphorylation, which basically leads to the synthesis of ATP, and some other ATP are also generated during glycolysis or during the citric acid cycle, what we call substrate level phosphorylation. Some of the electrons are also shuttled from the glycolysis into the mitochondrion, and basically these are the electrons which can affect the yield, the net yield of the ATP molecules by the glucose. We will discuss this later. So we know so far that these two molecules, NADH and FADH2, they're carrying electrons from food, from glucose basically. Now these electrons need to be transported to the electron transport chain. How does this process take place? So we will first talk about electron transport chain, and then we will talk about chemiosmosis, and we will see how mitochondrion synthesizes ATP. The electron transport chain consists of basically four immobilized or fixed protein complexes. This is complex 1, this is complex 2, this is complex 3, and this is complex 4, and two mobile electron carriers. So what happens basically, that we have studied that electrons are being carried by NAD and FAD. So NAD got reduced to NADH carrying electrons, and FAD got reduced to FADH2. So both of these two electron carriers have carried electrons from the citric acid cycle, and the job of both of these two molecules is to pass these electrons on to the electron transport chain. And this is our electron transport chain, which is composed of different protein complexes and is embedded into the inner membrane of the mitochondrion. So that's the outer membrane, and this is the inner membrane. So the inner membrane contains all these complexes and constitutes the electron transport chain. So these electrons, which were carried by NADH and FADH2, so they're shuttled from one complex to the other complex, and these mobile complexes, one is ubiquinone, and the second one is cytochrome C. So basically they help in faster, so they move rapidly between these complexes, carrying electrons from one complex to the other complex. So both of them, the ubiquinone and cytochrome C, so they will shuttle between the different complexes, and they will carry electrons from one complex to another complex, and ultimately they will pass these electrons on to oxygen, which is the terminal acceptor in this case, and this oxygen is converted into water inside the mitochondrial matrix. During movement of these electrons from one complex to the other complex, the transmembrane protein complexes, they pump protons from the mitochondrial matrix into the intermembrane space. So this is the outer membrane, this is the intermembrane space, and that's the inner membrane. So this space, which is highlighted in orange color, this is basically the space between the two membranes. So here is the outer membrane, and this is the inner membrane of the mitochondrion. So protons are not pumped out of the mitochondrion into the cytoplasm, rather they are pushed from the inner mitochondrial matrix into the intramembrane space, and pushing of so all of these complexes, they keep pushing protons into the into the inner membrane, into the intramembrane space of the mitochondrion. So this basically leads to a higher concentration of the protons into this inter membrane space of the mitochondrion, and these protons then fall back through ATP synthase. ATP synthase, uh, it's a rotary enzyme, and when they fall back, these protons, when they fall back through ATP synthase into the mitochondrial matrix, this leads to the synthesis of ATP from ADP and inorganic phosphate. So ATP synthase, it's a membrane bound enzyme which is responsible for the synthesis of ATP from ADP and inorganic phosphate. And ATP synthase can perform its job when protons are moved through the ATP synthase back into the mitochondrial matrix. So in principle, the protons keep cycling between the mitochondrial matrix and the intramembrane space. So these electron carriers, they shuttle these electrons through this electron transport chain. NADH basically works at a higher energy level at complex 1 as compared to FADH2, which is working at a lower energy level at complex 2. And this is the reason NADH would shuttle more protons into the intramembrane space as compared to FDH2, because this is working at a higher energy level as compared to FADH2. So these protons, they're pumped through these protein complexes, which are transmembrane proteins, and they're fixed. These four complexes, they're fixed into the membrane: complex 1, complex 2, 3, and 4. While two other electron carriers, they're mobile in nature: ubiquinone and cytochrome C. So they will shuttle electrons through the electron transport chain and the mobile carriers. And the job of the fixed fixed protein complexes is to shuttle protons. So mobile carriers are carrying electrons, and the fixed ones are there shuttling protons from the mitochondrial matrix into the intramembrane space. And when these protons fall back into the mitochondrial matrix, they basically rotate this ATP synthase enzyme. So this is a membrane bound enzyme. It's a transmembrane protein. It has got a channel, so protons can flow this through this channel into the mitochondrial matrix, and this helps ATP synthase convert ADP into ATP molecules. So, and the entire process is known as oxidative phosphorylation. It's a combination of two things. One is electron transport chain, so that's the electron transport chain, and second one is chemiosmosis, which is the synthesis of ATP resulting from the flow of protons through the membrane. So chemiosmosis and electron transport chain together are known as oxidative phosphorylation, or this leads to oxidative phosphorylation, or phosphorylation of the ADP molecules into ATP. So let's look at ATP synthase enzyme in details. As we can see, this is the intermitochondrial space, and this is the mitochondrial matrix, and that's the intramembrane space. So there are lots of protons which have been accumulated in the intramembrane space. The ATP synthase enzyme consists of two complexes. One is the FO complex. This is a membrane bound or membrane embedded proton channel, and F1 ATPase complex, this consists of the catalytic core. So that's the catalytic portion of the enzyme, and this is the membrane bound portion. The membrane bound portion, this also contains these channels for the movement of protons. We have previously discussed that protons can move through ATP synthase back into the mitochondrial matrix. So the membrane bound portion consists of a stator. This is the stator, and this stator has got two channels. One channel that is used for the entry of the protons, and the second one that is used for the exit of the protons into the mitochondrial matrix. So protons do not go straight through this complex into the, or through this protein, transmembrane protein into the mitochondrial matrix. Rather, they have to pass through these channels. So this stator is fixed, this doesn't move, and has got two channels. So protons will pass through the first channel, and now this this binds. So the next to the stator is a rotor. Rotor has got binding sites for protons, or for these ions, for proton ions. These proton ions, they will pass through one channel and bind to their binding site. So binding of the protons basically change the shape of each subunit, and this leads to the spinning of this rotor. So this rotor starts spinning by the binding of these protons, and each proton that binds to its binding site would complete one rotation before it can move out of the second channel into the matrix. So binding of one proton molecule to its binding site, and then this proton would complete one cycle during the rotation of this rotor, and then will come out of the second channel of the stator into the mitochondrial matrix. So this is how rotor is moved, and we call this a proton motive force, because this is a force generated by the protons, and helps this proton motive force helps rotor move in this circular fashion. The movement of the rotor also leads to the movement of an internal rod which is associated with the rotor. So the rod is associated with this rotor. So when the rotor moves, this internal rod also moves. This also rotates. Rotor rotates, and it rotates the internal rod. Next to the internal rod, we have got a catalytic knob. This catalytic knob is fixed by the stator. Stator keeps it in fixed position. So when the internal rod moves inside this catalytic knob, the knob itself doesn't move. This is fixed by the stator, but the internal rod, the rod inside this catalytic knob, moves, and moving or turning of the rod, basically it activates catalytic sites inside this knob. So this knob then can convert ADP into ATP. So this is how ATP synthase works, and this is also known to be the smallest molecular rotary motor in nature. So it has got two complexes. One is a fixed complex membrane bound, and the second one is this rotary complex. This is FO complex, and this is the F1 complex. So you have got one stator, one rotor, one rod, and one catalytic knob. Stator is fixed, has got two channels. Proton enter from one channel, bind to the rotor at a binding site, rotate it, and then they come out of the second channel into the mitochondrial matrix. This movement of rotor rotates the internal rod, and movement of the internal rod inside the catalytic knob, which is fixed, leads to the activation of catalytic sites inside this catalytic knob, and these catalytic sites can then become functional, and they can convert ADP into ATP molecules. So this is how ATP synthase generates ATP from ADP by the proton motive force. So what is basically proton motive force and how it works? So the proton motive force, basically we have got two types of gradients. One is a concentration gradient across the membrane, and the second one is voltage gradient. So one thing that we have got a higher number of some positively charged molecules, so we have more positive charge on the outside of the membrane as compared to the inside of the membrane. So you can consider this as a mitochondrial matrix, and that's the intermembrane space. So when you have got more, we have previously learned this thing as well. Uh, we have more positively charged molecules on the outside, so we have a higher concentration of that molecule, whatever it is, and we have a lower concentration of that molecule inside the membrane. So that generates a concentration gradient, which in this case is a proton concentration gradient. And second thing, because the molecules on the outside of the membrane, they're positively charged, so there's more positive charge on the outside as compared to the inside. So the inside is more electronegative and the outside is more electropositive. So putting these two things together, the concentration gradient and the voltage gradient, we call this generates what we call a proton motive force. So this proton motive force is generated by chemical energy. It is generated from chemical energy in case of mitochondrion and aerobic bacteria, what we have just studied, and it is generated by the light or from the sunlight in case of chloroplasts and photosynthetic bacteria. Now this proton motive force can perform lots of different functions. For example, we have just studied that this proton motive force is involved in synthesis of ATP. Excuse me. This is involved in synthesis of ATP. So when protons move through ATP synthase, they help ATP synthase synthesize ATP from ADP. Second thing is the transport of, where it works, is the transport of molecules against their concentration gradients. So you have to move the molecules against their concentration, so that means you would need energy. So you would either be utilizing ATP molecules to do this, as we have discussed previously in case of some carrier proteins, or you would need this proton motive force. So how does this work? So there are two different types of movements for these molecules. One is known as symport. In case of symport, when proton moves in one direction, it takes along another molecule. So movement of proton into in this particular case, for example, you want to move this X molecule against its concentration gradient into the sealed compartment. So proton would move through the membrane here into the compartment, and this will take along this X molecule. While in another case, what we call antiport, proton moves in one direction and the other molecule moves in another direction. So you would have a higher concentration of Y on the outside of this compartment and a lower concentration on the inside of this compartment, and you want to move you still want to move Y out of it. You want to kick this thing out. So it is not going to happen automatically because you have a higher concentration of Y molecules on the outside. So but this can be achieved by antiport. So proton would move through this transmembrane protein. Protons would move into the compartment, and the movement of protons into the compartment would push Y out of that compartment, and that's why this is known as antiport. So if the movement of the molecules is in the same direction, we call it a symport, but when it's in the opposite direction, we call it an antiport. Another function that is performed by the proton motive force is the rotation of bacterial flagella. So in bacteria, the movement of the flagella is also dependent upon the proton motive force, where protons move from one place to another, and they rotate the flagellum. So there are lots of other functions of the proton motive force as well. And one of them, the most important one that what we just discussed was the synthesis of ATP molecules. So that's the last slide for mitochondria. We have previously discussed the ATP yield per glucose molecule, and we have discussed that that's the maximum yield per glucose. You're going to generate 30 or 32 ATP molecules. And if we put these numbers together, we would achieve the same, like 30 or 32. But we have previously discussed that these numbers are approximate. Sometimes book even mention 34, 38. So what they're going to do, they're putting all of them together and um, uh, saying that you can produce 36 or 38 molecules uh, per 36 or 38 ATP per glucose molecule. But this is actually never achieved because we have discussed previously that some of the ATP molecules are lost because of the leaky membranes, um, and some of them are utilized in shuttling pyruvate into the mitochondrion. So the total number, it's a little complicated, but the total number of ATP molecules generated also depends upon the electron shuttles. So we would not go into details, but this is basically mitochondrial shuttle mechanisms. What happens that we have discussed that previously, if we can go back to the previous slide. I think it's not mentioned here, but some electrons are also carried via NAD in glycolysis. I think we didn't mention glycolysis here. We didn't discuss glycolysis in details, but when glycolysis takes place, NAD is reduced here. So means NAD is converted into NADH, carrying some electrons from glycolysis, like here. So when glucose is converted into pyruvate in the cytoplasm, this leads to the generation of some ATP molecules by a substrate level phosphorylation, and some electrons are also carried by NAD. So it means NAD is reduced now into NADH. Now the problem is this NAD or NADH cannot so this membrane is not permeable to NAD or NADH, so they cannot enter into the mitochondrion, but they have to transport these electrons that they have carried from glycolysis to the electron transport chain. So in order to perform this function, they have to they're dependent upon these electron shuttles. So there are two shuttles. One is known as glycerol 3 phosphate shuttle, and the second one is known as malate aspartate shuttle. We do not need to discuss in details, but basically these are two mechanisms which can help NADH transport its electrons through the mitochondrial membrane and to the electron transport chain. Because NAD or NADH themselves cannot transport their electrons directly into the mitochondrial matrix, so they have to pass these electrons on to some other molecules, and these molecules would then cross the membrane, and they would transport these molecules into the mitochondrial matrix. So that also depends which of the two shuttles were utilized for the transportation of the electrons into the mitochondrial matrix, and that can affect the overall yield of ATP per glucose molecule. But that's roughly the net yield, 30 or 32 ATP molecules. And as we have previously discussed, that the number is not fixed. You cannot precisely calculate what was the total number generated per glucose molecule because of the different things like leaky membranes, shuttling of pyruvate into the mitochondrion, and then shuttling of electrons from glycolysis into the mitochondrial matrix for oxidative phosphorylation. So some of the ATP molecules are utilized here, and the net yield is somewhere between 30 or 32 ATP molecules. So I think that is sufficient for today. Thank you very much. If you have any questions or comments, you can post them, and I would be glad to answer them. Thank you very much.